minute past noon. Let's get started. Good afternoon, and welcome to the NLM Colloquia on Biomedical Data Science and Computational Biology Research, whether you're joining us in person and or via the video cast. And for those joining us from the video cast, we welcome your questions after talk as well, and you'll see an email address in the slide. And if you don't write that down right now, the slide will be back after the presentation. My name is Zhiyong Lu, a senior investigator within the Division of Intramural Research at the National Library of Medicine. And before introducing today's speaker, I'd like to thank Dr. Richard Shorman, our division director, for his continued support for the lecture series, Samir Antani, and other colleagues, our speaker selection committee, our lovely admin Quinn, and training director Betsy for their efforts to make the events possible, and obviously our audio and fantastic audio and video team. Without them, they would be not possible. I also like to remind our trainees that there will be opportunity to meet with our speaker after the presentation to join for a lunch. We already have a number of trainees sign up for that, but if you haven't and would still like to go, we welcome walk-ins. Well, I can't promise there'll be enough free pizza, though I believe we do. I can promise you'll have a wonderful, fun, and engaging discussion with a thought leader in our field, and where no PI will be around. So today's lecture was actually scheduled originally in November last year, but we had to postpone because of the shutdown. Apologize for that, Dr. Shen. But on the bright side is that you are making two firsts. You are the first speaker of our lecture series in the new year. You're also one of the first speaker in this beautiful, newly renovated auditorium room. So now it's my great pleasure to introduce today's speaker, Dr. Li Shen. Dr. Shen is a professor of informatics and radiology at the School of Medicine at University of Pennsylvania, where he holds multiple leadership positions, including as the interim director of the Informatics Division, associate director of the Penn Institute for Biomedical Informatics, and the co-director of the Penn Center for AI and Data Science for Integrated Diagnostics. Dr. Shen is a leading expert in biomedical informatics, medical imaging genomics, and trustworthy AI large language model research. He previously served as executive director of the Professional Medical Imaging MICAI Society between 2016 and 2019, and he is an elected fellow of the American Institute for Medical and Biological Engineering, the American College of Medical Informatics, and a distinguished member of ACM and IEEE. Today he will speak about AI and informatics research for dementia and aging research. Without further ado, please join me in welcoming Dr. Li Shen.
Thank you so much to Dr. Lu for the very nice introduction, and I'm truly honored to be here, and also thanks to the Dr. Lu and the organizers for having me here. So, let me see. All right, so I'm going to talk a little bit about our recent work on AI and informatics, mostly for the dementia research and kind of aging care. I have like two parts. The first one, I'm going to focus on brain imaging kind of genomics. The idea is we want to identify genes, hopefully the targets for like Alzheimer's disease and related dementia. So this is one of the kind of the UO1 project supported by NRA, and in this work, we want to develop AI machine learning methods, right, to for like the AD discovery, mining the Alzheimer's disease biobank data. So So we have a few aims, right? Aim 1 is mostly to do the kind of genetic study, right? We have the whole genome sequencing data, then the AD kind of diagnosis, right? Try to find the genes that are related to Alzheimer's disease. Aim 2 is kind of we want to harmonize the phenotype data. There's a lot of, say, imaging phenotype and also the cognitive phenotype data, right? We can harmonize those, and in M3 we try to figure out the relationship between these, like the genomic data, imaging data, and the phenotypic outcome, right? And then in M4, using the AD biomarker we identified and also their genetic kind of molecular basis, we want to these could serve as targets, right? We're looking for a drug that can modify those targets. Yeah. Then the M5 is mostly for the kind of dissemination, right? And so this is part of this AI kind of ADSP Alzheimer's disease sequencing project, right? So ADSP has multiple consortia. So these consortia are mostly collecting the genetics and the whole genome sequencing data, right? And there's also a data harmonization consortium. So try to harmonize all these phenotyping data, right? And then we are part of the AI machine learning consortium. And so hopefully we can identify genes and proteins, and then there's a functional genomics consortium that can help to do the molecular validation, right? All these data are kind of available with kind of controlled access at this NIGATS platform, right? So we are analyzing the data available from that platform. And so Alzheimer's disease, just a little bit background. The kind of major challenge is there's no drug that can modify the disease, right? Although there are some recent kind of development, right? Now, a couple of FDA-approved drug available, but that can be used to treat a subpopulation of the patients, right? So people are still working on this. But we know the major hallmarkers, right? So there's amyloid plaques between neurons and neurofibrillary tangles within neuron, right? That's related to the tau protein, right? And also you can see the neurodegeneration patterns, right? And so we can see all these hallmarkers can be captured by like the imaging data, right? So there's amyloid imaging, tau imaging, and MRI and other modality can capture neurodegeneration. So we actually use these imaging as a phenotype, right, to study genetics. Ideally, we want to figure out the biological pathway from the genetic determinants, to the molecular or cellular signature, then the imaging biomarker, fluid biomarker, eventually they lead to the phenotypic outcome, right? Like a cognition and a behavior, right? So all these data are available, and so this is just actually Paul Thompson and I wrote a review paper quite a while ago. Yeah, but I think that the data, and we're still analyzing the similar type of data, right? Genomics, imaging, and outcome, right? And try to figure out their relationships. And then there are some interesting problems we want to study. So one is more like the heritability analysis of the imaging phenotype, so that we want to prioritize those imaging phenotype with a stronger genetic basis, right? And then once you have those phenotypes, you want to look for their genetic basis. Then these are the imaging genomic association study, right? And the other things people are studying is that we want to integrate genomics and imaging, right? Hopefully to better predict the outcome, or maybe try to figure out the biological pathway, right, from the genetics, omics, imaging to the outcome, right? I'm just going to give you a few example studies on each of these topics. The first one is on the more like the heritability analysis of imaging phenotype. If you want to extract, say, the meaning for like the imaging phenotypes, right? So sometimes we just use the atlas, right, to define those imaging phenotypes. It's more like a partition of the brain, right? And within, now you have different regions, right? For each region, you can calculate an average measurement. Could be kind of the average thickness or volume or maybe average, say, amyloid burden, tau burden, that kind of thing, right? But a lot of these existing atlases are defined by like anatomical knowledge or maybe functional study. They are not prioritized for imaging genetic study, right? So here, my previous PhD student, Jinshuan Bao, so he actually proposed a new atlas. We call that a GIANT, right? Genetically informed brain atlas. So what he did is we actually analyzed the UK Biobank data because they have both imaging data and the genetic data, right? And so there are almost like 40,000 subjects at that time available, then 7 million SNPs. And we use that to build the atlas and then try to apply that to a AD cohort, right? Alzheimer's disease neuroimaging initiative, and try to see that works. So here the idea is that we are using these kind of voxel-based strategy, try to estimate heritability for each voxel. But you have to align all these kind of different individual image into a kind of standard framework, register that into that so that the voxels are comparable across subjects, right? So we use this Ravens map to do that, which was developed by my colleague, Christos Stavazikos. So now these are the gray matter. You have this number of voxels, and the white matter, so this number. So there's a lot of voxels, and we did a more like voxel-wise heritability analysis using this LDAK tool, right? After we have this more like a heritability map, right, in the brain space, then we can do the segmentation. So Jinshuan proposed a 3D Gaussian mixture model, right? So here's the model. The idea is like, the voxels with similar heritability estimates tend to be grouped together, right? And also voxels are close to each other, should be close, kind of clustered together, right? So based on these, then we developed this model. And so after apply our model, then we get a segmentation or something like this, right? And that will become our atlas, right? So the results, if you look at using our segmentation, then for each region, within the region, the voxel level heritability are more homogeneous, right? And another thing is so you can actually, based on our region, you can calculate some region level phenotype, right? So certainly we apply this to an independent data set, then so there's no circularity. But using different atlas, the GIANT, like a watershed, is just a different algorithm applied to our voxel-based heritability map. And the Muse is an existing atlas. So we can show actually our regional measure are more heritable, right? And if you do some GWAS or other genetic analysis, we can identify like more hits using our atlas. And then we also kind of showed the genetic landscape of these giant traits. So here are just start from the raw image, and then we do voxel level kind of heritability analysis. You have the map, then we do the segmentation, right? Then we did some like validation, evaluation, right? Because you want to generate that using different data sets, right? So that the results are comparable, similar to each other, right? And so then we also did some evaluation. I showed the results in the previous slides, right? Imaging genetic analysis. We can show actually these phenotypes are more kind of heritable, and we can identify more hits, right? And finally, the atlas. So here are some like results. These are the voxel level heritability and the p-values and the map. And so we extracted 50 giant regions, right? And so here is the landscape of the giant region. Kind of, we have the chromosome. Basically, we did this imaging genomic analysis, right? Here are some findings. Different color corresponds to different brain region, and these are different chromosomes, right? Then you can compare the, say, genetic basis between our giant region and also some like typical phenotype, like weight, height, BMI, right? You can see they share some genetic basis, right? And these are just some functional annotation. And so here are, for these giant traits, right, we can calculate the genetic correlation and also their phenotypic correlation. They are similar, but not exactly the same, right? So hopefully, in the future, we can use all these traits, right, to have some interesting imaging genetic findings, right? So that's the first work. And the second work, I want to discuss some approaches to identify the imaging genomic kind of association, right? So once you have the imaging phenotype, then we want to look at their genetic basis, right? So this is more like a very classical work we did like a long time ago, 2010. So one of the first brain-wide, genome-wide study in Alzheimer's disease. So we have all these imaging phenotypes, right? These are regional measurements, and then we did just a pairwise analysis, GWAS of all of those, and we can summarize all the results in this heat map, right? So if you are interested in seeing a genetic effect of certain SNP, then you can map it onto the brain. You can see which region are affected. And if you are interested in certain, say, brain phenotype, then look at the GWAS, right? So the Manhattan plot, so we can see some of the hits, right? This is the hippocampal gray matter density. So it turns out we identified some very interesting findings which are missed by traditional, say, case control GWAS, right? And our sample size is much smaller, and but we can still detect those signals. That shows the promise of the imaging phenotype, right? So that's statistically more powerful, right? So that's why that's the feature of the quantitative traits. So in addition to these, say, the imaging traits from the structural MRI extracted from, we also looked at some other quantitative traits, like the cerebrospinal fluid biomarker, right? And then the amyloid imaging, maybe the memory scores, cognitive performance, or longitudinal amyloid. So we kind of identified a lot of interesting findings. So some of those, like this one, has been used in the model AD to basically as build a knockoff this gene, build a new mouse model. So then that will be very valuable for the subsequent analysis, right? So that's the traditional, say, statistical analysis. But people are also interested in some, maybe if you are a computer scientist, you want to extract the new features, right, from the original, say, the SNPs, right? People are calculating, say, polygenic risk score. So this is a weighted sum of these original genotype value, right? But in the imaging domain, you can also calculate a weighted sum of the original imaging measurement, right? So people have been using a canonical correlation analysis, which is very popular in imaging genomic studies. So try to maximize the correlation between this genetic component and the imaging component, right? So that will be a new finding, right? And but in order to reduce the risk for overfitting, you have to add some additional constraint, right? So to kind of make the model not that flexible. So typical approach is you can use lasso, right, to add this sparsity constraint. In that case, not all these original, say, SNPs are relevant. So some weights, most of the weights are zero. Only non-zero weights correspond to relevant genetic marker, right? But the imaging marker, you can also do the same. And in this work, we actually add some additional knowledge. We call that we use this preference matrix to store that. So say if you already know some association between a SNP with some imaging phenotype, you can mark that, right? So with this prior knowledge, and we developed a new kind of constraint, the CCA model, right? So here are some, just a proof of concept study. So we have some phenotype data, right? And in this case, some are, we know actually there are AD biomarkers. Others are just, say, noisy patterns. We just extracted some from the gene expression data of the plasma, and they are not related to AD. And then we have some AD SNP and some randomly selected SNP. So we apply this model. So these are just shows some of the data, right? But it turns out compared with traditional CCA and sparse CCA, our preference matrix guided CCA and sparse CCA, it works better, right? In terms of the correlation we detected, on the training data, I think they are kind of comparable. But if you apply that to the testing data, so these traditional model, actually, there's no signal at all, right? It's zero, the correlation. But here we still have some signal. So, and if you look at the relevant features, so here we have the CCA, sparse CCA, these are our methods, right? And we have the genetic data and imaging data. So, and we know some of these things are actually not relevant, right? It's in the middle, the zero. So our model can detect relevant signals, but the existing model, like, they are mixed together. So this is actually just an example of the imaging genomic analysis. And there's a large body of the kind of the literature on different methods. Yeah, so I think if you're interested, you can take a look at that. Here I just show you an example. And the third one I want to talk about is more like the data integration, right? Basically, we want to figure out the biological pathway, right? From genetics to omics to imaging to outcome. If you see the evidence at different scales, different levels, the results might be reassuring, right? Because we can have multiple kind of levels. We have the same signal. So that's what this is also done by Shen. So what we did here is we are also using the UK Biobank data. Then you can do imaging genetic analysis. So there's a lot of findings right here. And we also have these case control study, right? You can just look at, say, a lot of like Nature Genetics paper in AD genetics. So we can look at their summary data, and we can align their GWAS result with the imaging kind of GWAS result, right? So here you have two phenotypes, right? So then we want to see whether the signals are co-localized, right? So now if we are looking for this type of pattern. And here these are two phenotypes. That's not the one we are looking for. Then based on the co-localization analysis, we can identify a kind of a subset of these things. And another thing is, like in UK Biobank data, we actually have the individual level data. Then we can do further mediation analysis on those subset of the findings. Then we actually identify a kind of a imaging measurement in the entorhinal area, which can serve as a mediator that mediates the genetic effect of this SNP onto the dementia outcome, right? Then we have this finding, but we're still missing the omics level, right? But there's a lot of like existing database, like the GTEx, right? They have EQTL result. Yeah, we can also do the co-localization analysis to align this with the imaging genetic result. In this case, we actually identified a gene which happened to be kind of the SNP we identify is the EQTL that can regulate the expression level of this gene. And this gene is highly expressed in this cell type, illegal dangerous sites, right, cell type, and also in this region. So in that case, we can connect all these dots together, right? So that's probably I skipped that. These are just the pipeline, the analysis pipeline. And but here are some biology, right, about our findings. Yeah, I won't go into the detail, so my postdoc, Tianhua Zhai, so he's very interested in kind of study this as a therapeutic target, right? Yeah. So he has some previous work based on the base one. So we know this is the target people have been studying kind of extensively, but a lot of all the clinical trial failed, right? But the thing here is that we want to apply AI and machine learning. So the failed trial only tested a few compounds, right? But there are a lot of, say, FDA-approved compounds. So here we have a lot of data available, right? Maybe here we can learn a model and then try to apply that to the new compounds and at least first estimate the binding affinity with this target, right? So then we can just to pick a top-ranked one for the subsequent analysis, right? So that's actually what Tianhua did here. So here we're looking at you can do the molecular docking simulation, right? Extract some docking features, right? And also on a subset of the compounds, right? And then we can also look at some kind of other structure, right? Features like the QSA feature, the quantitative structure activity feature, right? So we have all these features, and then in this work, we kind of did an analysis to do an individual kind of analysis for these modality and also combine those together. So you can see we tested some simple machine learning models to predict the binding affinity quantified by this IC50 score, right? So we can see it has some signal, right? So the R square is like 0.6, and the best one is like here. So it has some signal, but there's a lot of future work we want to do. Maybe we can develop more powerful machine learning models, right? Yeah, basically that's all I want to cover for the first topic. So yeah, this is a review paper. Actually, Shen did. It's kind of a year ago, right? More than a year ago. But if you are interested, yeah, so we summarized some informatics strategies for analyzing all these genomics, omics, imaging, and phenotype data. So here just a brief summary on this topic. So we looked at 3 topics, right? And we're also interested in doing some like single cell analysis. So this is my PhD student, Zheshen Wang. He did something, but not on the AD data. So it's a single cell analysis. But the thing is, since we study like the individual, right? So for the single cell data, all these gene expression for all the cells, how can you come up with kind of a sample level similarity, right? That's what he did. He developed some method using the optimal transport. So we can calculate the similarity at the sample level using the single cell data. And another thing is I'm very interested in kind of study the immune-centric, like the AD kind of the target also. So in this case, actually, so another student, so Zhongchen, so he actually did some for the immune profiling. So at Penn, we have Institute for Immunology and Immune Health, and they are doing immune profiling, try to build something like UK Biobank data, right? Biobank, and then we're just doing the, say, cytometry, right? Profiling for the patients visiting, say, Penn Medicine. So they have a lot of these cytometry data, but the thing is, in order to analyze those, right, you have to do the manual gating, which is very time-consuming, and you cannot do that in a high-throughput fashion. So John actually developed an automated kind of machine learning, a deep learning framework, so that you can do this automatically. So this is some initial work published, but he's still trying to figure out the kind of improve the tool he developed. Hopefully in the future we can deploy that on certain platform. Yeah, so that's something. And in the future, hopefully we can also apply that to the AD study, right? So yeah, that's the first topic. Then I'll briefly talk about the second topic. Mostly I want to talk about some multimodal AI and generative AI for dementia research, and also supporting the, say, aging caregiving, right? So this is another UO1, actually, so between Jason Moore, Marilyn, and myself. And so in this work, we try to also analyze the omics data, imaging data, but try to incorporate the knowledge base, right? And develop the AutoML system so that we can, the AutoML system can identify maybe a data set and also algorithm, then generate a result, what you are looking for. So in that case, the user does not need to be a machine learning or domain expert, right? It's like an iPhone, right? Everyone can use that, right? This is kind of similar to this proposal was written probably 5 or 6 years ago, right? But this is kind of aligned with all these new development using the foundation model, large language model, and AI agents, right? Or knowledge graph can serve as a retrieve augmented generation, right? So basically, the agent can do the decision making without any help from human, right? So That's kind of the many system developing nowadays are kind of want to achieve the same goal. Yeah. But for now, I just briefly introduce some of work we did before. So since we start from the AutoML system, so Ray actually is in Jason's kind of the department. So he developed a Streamline. That's an AutoML system. Some of my students, trainees, they kind of contributed to certain algorithms and implementations, right, in the Streamline, and these are for classification, so these are the regression, and we also use that to analyze some Alzheimer's data, right, have some interesting findings, and we're also developing like a new machine learning methods, right, so like in this one, hopefully can incorporate that into the library of all these the tools, right, so for, say, in this case, for disease classification, here the innovation is basically if you misclassify some sample which is close to the boundary between, say, control and the case, we shouldn't give them a lot of penalty because it's a difficult case. But if you misclassify something which is very far away from the boundary, we give a large kind of penalty, right? So that's the intuition behind this method. So we have the, say, these are all healthy people, right? So that's their distribution, the values, right? So these are the x-axis are just the values of the predictor. But these are our predicted value, right? We can calculate some kind of Wasserstein distance or maybe the optimal transport, right, between these two distributions, right? In that case, since far away, we'll get like a more penalty, right? Since close to, say, the true things, although it's misclassified, we'll get a less weight, right? So that's the intuition. So it turns out it works pretty well. Right? You can see our approach actually works the best compared with a lot of existing methods. And another thing we're interested in is, in addition to doing the predicted outcome, we also want to align things together so that we can kind of do the progression and the score estimation and also maybe the staging, right? In this case, we can use AD biomarker, right? And we use some diffusion model kind of implemented by a tool called the FATE, right? So we can actually map all these data, right? So each row is a sample and each column is a biomarker, right? Then here in the new embedding space, these are FATE dimensions, right? You can see actually here are healthy people, MCI patients, and AD patients. They form a linear trajectory, right? So if you fit a curve, this could be the pseudotime or the progression, right? So that will be very nice so that you can align all the biomarker against this pseudotime, right? You can build a certain profile. And we're also comparing this with another tool, like the Sustain. They can also do the staging and even the subtyping. So we can see they actually would agree with each other pretty well. So here is the FATE result, and we'll color map the diagnosis, right? So it's aligns with the diagnosis. This is using a different data set. The previous one is using all the AD biomarkers. Here we are only using the amyloid imaging, right? And so these are using the volumetric, right, MRI measurements. These are the tau imaging. So here you can see the Sustain result. They have the staging, right? It actually aligns with the FATE result very well, right? Especially for the amyloid. And for MRI, not that well, right? But the tau, kind of, yeah, some kind of. So we're still actually working on this. Hopefully can improve this and maybe add some prior knowledge to better predict, kind of generate this embedding. So these are just a different outcome, right? So one thing I want to talk a little bit about is the interpretable AI, right? So we have been working on developing methods and also apply that to different applications in AD and ADRD. Try to make the machine learning model and interpretable, because so that the doctors, if they want to use your model, they need some additional evidence, right? So why this result is true, right? So there are different approaches, right? So here I just list a few and just give you a few examples, right? So since we have this black box, right, you want to understand what's going on inside. The low-hanging fruit is you can look at the input level, right, the features, which feature are important, or you can look at the output level, right. maybe there are some concept can explain the output, right? So this is one study we did is we want to predict the survival outcome, right? So here, but at the same time, here we can predict the time to event prediction, right? So here's the risk trajectory, right, over the time. So here's a probability, yeah. But different people, they may have different risk, right? So we can do the clustering at the same time. In that case, if we predict the trajectory, but at the same time, we can also explain whether this belongs to the low-risk group or high-risk group. So that's certain like explainability, right? So that's why we call that interpretable subtyping, right? Another thing is at the feature level, you can map. We have a very simple network, right? You can either map the weights to the original feature. You can see which feature are relevant, or you can use methods like SHARP, right, to measure the feature importance. So these are explainability. Now here in this model, we actually build some expert distribution, and then you just for individual patient data, then you can estimate the weights. So the output trajectory will be weighted sum of these expert distribution. It turns out the expert distribution we learned using that weights, you can group different subjects into different clusters, right? And the cluster we identified is actually matches with the Kaplan-Meier plot. So you can see these are the expert distribution we identified, right? You can use this to explain our outcome. So if you look at the clustering result, our method is the best. But these are the kind of the prediction accuracy, right? Try to see the predict trajectory whether matches the actual data. So ours are not the best, but in one case it's the best, right? But the goal is here, the major goal is try to do the clustering, right? So yeah, so that's two examples. And another one is this is the NAM is neural additive model, right? It's also at the input level, because if you don't want to use the feature directly, you can actually build a network for each feature, right? So you can learn a network. And here is the contribution to the final prediction, right? And you can build a shape function. So x-axis is the value of the feature, and the y are the contribution to the outcome, right? And so if you see a wider range, that means the range of the value does make changes for the contribution, right? So this is an important feature. So we actually added this, so you can actually rank all those features by looking at this shape function. So these are some identified features, and we compare with some other methods. We can see, so the clustering, we can actually still get the best log rank result, and for the prediction, we get a pretty competitive C index, right? But this model is also interpretable. And the next one is on the output level, right? So the output level is the concept bottleneck. I guess probably many of you have been using these. Actually, we are using the mimic data, the clinical notes, try to predict the dementia stage, right? Say ask GPT or what's that, then we don't know, even though the prediction is correct, right? So we don't know what's going on, right? It's hard to explain. But what we did here is we try to mimic the doctors, right? There's a medical textbook, how doctor diagnose Alzheimer's. The GPT to extract some concepts, right? Maybe there's a cognitive decline, or maybe there's a memory loss. These are different concepts, right? Once we extract those concepts, then we can use kind of here, when you do the inference, right? You can check whether these nodes kind of can generate a concept. This concept is yes or no, right? So basically, you can say, oh, based on these nodes, whether the patient exists cognitive decline, right? So in that case, we can create a concept vector. Then you just use the concept vector to predict the outcome. Then based on the outcome result, you have which concepts are relevant, right? So this is concept bottleneck model. You can see, so you have all these concepts, and some of the feature is absent, and others of feature is present, right? And our results is actually also outperform some benchmark results. So that's another way to do that, right? The other things are like the mixture of expert, right? So here we can build different experts. Each expert is in charge of certain region, right? We have all these regional measures, but you can group those into different, the intermediate kind of larger regions, right? So, and then we have this MOE method. Then you can actually, it will give a weights to each expert, right? And you can map those weights to the original, let's say, the brain space, and can see which region are relevant. So these are also some explainability result. And the final one is like a large language model, right? So here, since a lot of data we are dealing with are tabular, right? So we know the language model is to handle the language data, maybe the sequence data, and there are also model to handle the vision, right? The image data. But a lot of data are tabular. So here we actually want to build a kind of a language model for the tabular data. So Sophie and Shu did this. So based on existing, like it's called a table GPT, and what we do here is we have the tabular data, and you can formulate our problem as in-context learning, right? So here I have all these data, I know the label, but this data I don't know the label, and I can just ask whether what is the label should be, right? And we can do some fine-tuning, right? Given if you have some training data, right? So here we can fine-tune the model, and then here we just do the inference, right? The model parameter are frozen, right? So, and it shows actually compared with other language model, our language model works the best, right? And another thing is you can also get something behind that, right? The reasoning. So that's the explainability. So here, if you do the zero shot, then you compare with other language model, ours is the best. And so these are also language model. But if you do the in-context learning, you can also use that to the traditional method, right? Using other examples to train a model. Then still our kind of method is the best. But if you add the reasoning, so here you can even improve the performance, right? Without the reasoning, because you can ask, say, what's the reason behind that? So it will provide this. So this actually is a pretty promising, right? So yeah, so that's some interpretable AI work, and so I guess I don't have a lot of time. I just very briefly go over the remaining slides, right? So here is we kind of want to evolve the large language model and the knowledge graph, right? We can say, we can actually use large language model to query the literature, add a new entity to the knowledge graph, right? You get enhanced the knowledge graph. And at the same time, so we can use actually the knowledge graph to do RAG type of analysis, right? Try to avoid the hallucination, right? So because these are some existing evidence, right? So that's what we did here in this work. So it shows actually pretty good. And another thing is, since we can use large language model to add entity into the knowledge graph, in this case, we actually add some non-medical factors into a prime KG, or existing knowledge graph. And we can show actually, after doing that, you can, if you do the link prediction, you can get better accuracy. On the other hand, we actually did some discovery. So you can actually identify some genes that are connected to the AD node, and you looked at some additional single cell and multi-omics data. We can see actually their correlation. So that's another work. And this one is also for the large language model. You can even use that to do the feature selection, right? And here we're analyzing the genotyping data, and you can ask a large language model which feature are relevant, or whether you want to combine a different feature together. So you have the feature extraction and also feature engineering. So it shows some promising results, right? And finally, I want to, so this is another project. So we want to identify the AI and the technology to help caregiver giving like a support for either like the elderly people, healthy elderly people, or maybe dementia patients. Yeah, one study we did is to support the caregiver, right? So they have mental condition, their behavior intervention. We actually recorded that and analyzed the transcripts. And in one work, actually, we can build maybe a chatbot, right? So then pretended to be the therapist to provide a kind of feedback, right? And we actually even shared our data in the public domain after denormalization. So this was published in KDD. Other things actually, we are analyzing the transcript to predict the burden, right? And also, this is a new project. Actually, the NSL project basically want to use the EHR data, right? Maybe to predict some like event, whether the event of, say, the risk for injury will happen for the dementia patients, right? Developing AI and machine learning models. So this is just some analysis to analyze the transcript data, sentiment analysis. Then we can extract relevant, say, the conversations that can help the future, like the therapy, right? This is the kind of the conversational thing. We shared this data, and we show actually using this data to fine-tune some existing large language model that can improve the performance, right? Yeah, so I guess basically that's all I want to share. So this is just a summary. I talked about 2 topics, right? These are just some subtopics, and yeah, so these are the kind of the focus of my lab, and so yeah, basically we're developing informatics, computing, and AI machine learning methods for dementia and aging study. And finally, I want to thank NIH, NLM, and NSF for their generous support to support our research, and also my previous and current lab members. Yeah, I guess that's it. Yeah. Thank you so much for your attention. Oh, okay, great.
All right. So this is now the QA session of the talk. So we'll take some questions first from the audience here in person, and then if there's anybody online who would like to ask questions, please submit your question to the email address shown in the slide. Anyone?
So the microphones seem to work.
Oh, yes. Yeah, you need to. Yeah. Thank you, David. So before you ask a question, please press the button, mic button. Richard.
Yeah, really nice talk, a lot of impressive work. At the beginning for the GIANT project, one of the goals was to identify phenotypic endotypes of Alzheimer's disease. So I wonder, were you able to identify those, and what are the features that distinguish the different phenotypic endotypes?
Yes, I think we did some kind of analysis on the, so as I mentioned, we use UK Biobank to generate this atlas, right? And then we applied the atlas to the ADNI data, so which is the Alzheimer's Disease Neuroimaging Initiative. And so we did some GWAS study, and so using these kind of giant phenotype, we actually identify more hits. So yeah, some are actually new discovery. There's some discussion in the manuscript. Yeah, so probably in terms of biology or.
But like, what are the different types of Alzheimer's disease, and what are the genetic or the structural differences between the different subtypes of Alzheimer's disease?
Oh, yeah, you mean the subtype?
Yeah,
right?
the endotypes. Yeah.
Oh, yeah. When I say the endotype, it's actually not a specific subtype. Yeah, I know there's some terminology. So the endotype is just the imaging measurement. Mm-hmm. So okay. Say if I have an atlas, right, and it's just a partition of the brain. It partitions the brain into different regions. Mm-hmm. And for each region, I can extract some relevant features. It could be, say, the amyloid burden in that region, mm-hmm. or maybe the volume of this region. Or maybe if it's a cortical surface, it could be the cortical thickness or curvature, right? Mm-hmm. So those measurements, we define that as endophenotype, okay. But not the actual subtype.
Yeah, okay, yeah. Okay, thanks.
Right, yeah.
I do have a question from online audience. The question is, markers such as amyloid deposits in the brain, especially the ADIN cohorts, do not have a good correlation to patients' symptoms. can your PET/amyloid/tau AI model combined with the brain atlas be used to predict future AD in aging non-symptomatic patients with amyloid and tau deposits in PET imaging?
Yeah, I think that's a great question. So this is kind of aligns what we see in this trajectory analysis, right? So if you look at all the biomarkers, right, then you can kind of reconstruct the trajectory in related to the diagnosis. Because AD is not a single disorder, right? So when you look at the diagnosis, it's a mixture of all the subtypes. If you use, say, only the amyloid, right, then maybe it only captures the amyloid subtype, right? And there's a lot of other subtypes that are mixed together, so they may not be amyloid positive, right? In that case, it kind of like reduces the predictive power of using amyloid alone, right? So, but yes, so Dr. Lu mentioned, so basically if you integrate all these modalities, I think there's a potential you can better predict the diagnosis. It depends on your goal, right? If you just want to make diagnosis, that's a good thing, right? So, but another thing for the subtyping is if you want to develop a drug, right, probably you don't want to use diagnosis as phenotype, because I want to find a kind of a homogeneous mechanism underlying certain subtypes so that I can identify the actual target, right, and look for drug that can modify that specific target for that specific subtype, right? For different subtype, you may need a different drugs. That's why we need to do the precision medicine, right?
Yeah, so that brings me to the question I was thinking about, is that ideally what you would, and I don't know how far along this is, when this will be possible, but ideally what you would like to do is to have a pathway and be able to extract out the pathway and then treat different parts of the pathway for clinically. How far away are we from being able to understand, using the technologies that you have, some of these pathways for AD or any other disease?
Yeah, I think that's a great question. Yeah, definitely people are working on these towards these directions, right? So that's actually one of our major focus. So I want to do these kind of trajectory analysis, basically using data-driven approach, right? If I have all these data, then I want to find a kind of an embedding space, a new feature representation. And in this space, we can figure out not only the trajectory or the staging, right, but also the subtypes, right? Because the healthy people could be here, but one subtype probably it's over there in the feature space, based on the similarity of these patients, right? If patients are similar, they probably, their trajectory, if they are similar, they tend to belong to the same subtype, right? Maybe you can one subtype goes to here, and another goes there, and another goes there. But all these are the AD patients, right? The diagnoses are the same. But if we can kind of deconvolute all these trajectory, then I can identify these subpopulation, right? And then we can do the genetic molecular analysis for the drug development, right? And another thing is, based on that, that can also help the diagnosis or the staging, estimate the severity, right? And also determine the subtype for the precision medicine purpose. I think people are working on this. Yeah. So, but AD and dementia are very complicated disorder, right? That's certain kind of progress, I would say, right? Because at least we have some new drug development, right? The new drugs are approved, and but that can only treat a kind of a subset of the people. But people are still working on that, try to figure out maybe new subtypes, new targets, new drug, right?
Thank you for this multidisciplinary study. There's a paper that just came out by Chaubet on possible reversal of Alzheimer's disease related to NAD+, And can you comment on that paper? How does it relate to your work?
Oh, yeah. What was that paper? So what's the discovery?
A paper by Chaubet on possible reversal of coming back. Well, the disease becoming less strong for Alzheimer's disease by Chaubet. It just appeared.
Actually, the title is Pharmacologic Reversal of Advanced Alzheimer's Disease in Mice and Identification of Nodes in Human.
Oh, in the mouse model, right?
Yes.
Yeah. What was the cause? Sorry, I didn't get that. Yeah. So the reason is they found the cause.
Yes.
Yeah. What was that?
They
reversed, right?
Yes, they reversed it.
Oh, so you can. It's kind of like a disease modifying mechanism, right? Is that? So did they do some intervention to reverse the process? Okay.
Yes, but
okay.
We did not see where NAD plus or respiration was coming in the slides.
Oh, the image and.
Yes.
So sorry.
Is it a new paper, just by chance?
It's a paper in Cell Medical Reports yesterday.
Oh,
yeah,
it was
so
yesterday, and also they have some intervention, right?
Yes.
So the reverse, did they use imaging or other things to quantify what do they mean by the reverse, right? So they might see some performance improvement.
I guess it's so new. What I would suggest is we send the papers to the speaker.
Yes.
Yeah. We can discuss offline. Yeah. Sorry about that. Yeah.
Rich, you have a question?
So a lot of times it's useful to do dimensionality reduction before you feed your features into these complex models.
Yes.
And one way to do that is to be able to do topic modeling, where you basically group together features that have correlation structures and things like that.
Right.
Have you tried to do anything like that, to do the dimensionality reduction via topic modeling?
We probably haven't done that by the topic modeling. But I think we did do a lot of these things are dimensionality reduction, right? Yeah. So if you map the original representation into a new space, that space, if it has a fewer kind of dimensions, so that's dimensionality reduction.
Right.
I did show you we have some like using language model to do the feature selection and the feature extraction, right? So we didn't do the topic modeling specifically, but you can use the language to ask the language model whether now we have a list of all these features, which is genotype data, so a lot of features, and which features are relevant, right? Because behind the language model, they have all this knowledge available, right? So if they think these are relevant features, then based on our query, so they will give us some result. I will actually have a procedure to do that because you don't want to do that once. So we did that more like bootstrap or a lot of things and select some reliable features. And it shows actually it works. It works in some cases. Yeah, we did like two analysis, especially for estimating the, I think, genetic ancestry. Yeah, that works. Yeah, and other things that you can also even like combine different features together. You can also ask a language model, right? But doing the topic modeling, yeah, I think that's a great idea. So probably we will think about that. Yeah. Right? The thing is you can group relevant features together, right? So say all these features belong to the same topic. I can either do a PCA, capture everything, or if they're highly correlated, maybe I can pick one of those.
Right, exactly.
Yeah, definitely.
I mean, one of the advantages of that is in separate studies, maybe you have overlapping features, but if you know which features correlate, you can actually merge them into a topic, and then you get the topic across the different studies.
Exactly. These are some kind of high-level thing, right? So say, if you can see some high-level signal, I have all these genes, but there's also pathways, right? So pathway can capture some collective effect, right? Yeah. Yeah, great idea.
Time, but I want to take one more question, last question from the audience. Go ahead.
I have a question about staging, like the staging and zero time trajectory. As you saw it for AD progression, So zero-time method assume primarily a monotonic trajectory, as we know. So my question is, like in Alzheimer's disease, different biomarkers peak at different phases. So I feel like, how does your model handle non-monotonic or multimodal, multi-phase trajectories? So how do you handle that?
Yeah, great question. Yeah, I think that's exactly what we want to do, right? To try to figure out not just the trajectory, but also different type of trajectory, which may correspond to different subtypes, right? Because for different subtype, the order of the abnormality for all these features may be different, right? You can use that, the ordering, to group different patients together, right? So say, I'm just so say for the amyloid, right? So amyloid positive, maybe they see amyloid become abnormal first, right? But if a different pathology, they see a different kind of the measurement become abnormal first. Then the ordering between amyloid and a different biomarker may be swapped, right? Then you can actually use that information to group different subjects into different subtype, right?
All right, thank you.
All right. Well, thank you so much for the wonderful talk, and thank you all for coming.
Thank you.
Again, just a reminder, for the trainees who signed up already, you know where to go. For those who decide to join but have not signed up, just come to see me. Thank you.